Hi everyone, I'm Annie. I'm here to give a webinar on LAMP PCR. So, what does a LAMP PCR mean? A LAMP PCR stands for loop mediated isothermal amplification developed by Notomi. The LAMP reaction proceeds at a constant temperature using a strand displacement reaction. LAMP reactions have found their way of great importance in molecular diagnostics. So, how effective this LAMP PCR is that it is used as a nucleic acid amplification testing procedure. It uses DNA for amplification, or you can use for RNA detection using an RT reaction as well. So, what are the characteristics of LAMP? The LAMP reaction is very simple, it's specific. The reaction doesn't require any denaturation step, it's isothermal. It is very specific because it has been designed with 4 to 6 different set of primers. It's rapid and it's cost effective. So what happens in a LAMP reaction is purely based on the LAMP primer designing. So the LAMP primers, they have 3 sets of primers, that is the internal primers, external primers and loop primers. So let's move on to the principle. So what happens during a LAMP reaction is that a target gene has been incubated with few of the reagents like the buffer, the DNTPs and the BST1 enzyme at 60 to 65 degrees centigrade. Okay, as the reaction for 30 to 1 hour. So as the reaction progresses, you can see a color reaction, that is a visual detection can be seen whether the products are being formed or not. So what is the principle behind it? What are the stages that this undergoes? So the step 1 happens as follows. So the first step is the LAMP primers anneal to the complementary sequence of the double-stranded DNA, which initiates the strand displacement activity. So like the other PCR reactions, they do not require any denaturation step. So it directly, the primers go and anneal to the target DNA. So internally, the outer primer as shown in figure, FIP, binds to the particular complementary region and initiates the strand synthesis. So again the second step happens. The outer primer F3 hybridizes to the F3C region and the strand synthesis again starts. Now similarly at step 3, the third primer initiates its reaction forming the complementary loop. The forward as well as the reverse primers start displacing the double-stranded DNA and starts forming the products. Meanwhile, the primers internally complement them, forming the loops on the outer ends. So as the strand synthesis comes to an end, the loop primers starts detaching and letting loose the synthesized strand. So similarly, in the subsequent steps, as the primers keep annealing, the loop formation is formed. is the loop formation is in place. So as the loops keep forming, the products keep detaching from the synthesized strand. So the synthesized strand forms as the template for the further reactions. So I have demonstrated a gene which clearly shows the F3 region, F2 region, and the loop region. So how the primers are being synthesized. So each step, as the lamp keeps forming, the target sequence is amplified so many folds, and the reaction completes with lot of loops in them. So the loop primers helps to achieve the maximum amplicon in a short span of time. So, as the figure depicts, the lamp detection happens in a tube. Usually, a dry bath is preferred because of the cost effective machine than a thermocycler. So, the reactions are set up using an Eppendorf vial with the color detection or the fluorescence dye as per our choice. So the reaction which I have shown has cyber green in it. So against the UV light, this gives a fluorescence. So the negative control is not showing any fluorescence, whereas the positive control is showing fluorescence, which means to say that the patient is positive of a particular disease. So what are the advantages? Amplification of DNA takes place at an isothermal condition and the efficiency is very good. The thermal denaturation is not required. Specific amplifications can be seen. It's highly cost effective. Reverse transcriptase is like a positive thing for lamp and takes less time for detection. The disadvantages of this reaction is that the primer designing is very complicated and it is sensitive to contamination and small changes in the condition. So it has found its rapid diagnosis at viral and bacterial and parasitic disease. It helps in the identification of genus and species specific parasites. So the efficient primer designed for loop mediated isothermal amplification using a lamp designer. That's the best software to be used, and blast results of the particular primers can be checked. So how do we compare the lamp with the PCR? So as the table says, the lamp PCR is an isothermal reaction, whereas the polymerase is a cyclic reaction. Here in lamp, the isothermal temperature is around 60 to 65 degree, whereas in the PCR it is variable and you need the subsequent denaturation, annealing and polymerization reaction steps. It doesn't require expensive thermocycler, it requires a thermocycler. So detection is greater in loop mediated, whereas in PCR the detection is lower. Amplification specificity is higher as it uses 4 to 6 nucleotides, oligonucleotides, sorry. Amplification is specificity than the specificity is lower than that of lamp. The visualization of DNA could be done through through the normal visible light or by using gel electrophoresis. Here in the normal PCR you cannot see the DNA visualization. Could be done using crude DNA samples. Here you need purer DNA samples for specific amplification, and loop primers accelerate reaction rate, no loop primer in the polymerase chain reaction. Thank you.